All right, so we might kick off now. So first of all, welcome everybody to the first of Genomics England's research seminars for the year. So we've got two excellent presentations coming up today. One from internal within GEL and one external. And so as usual, if you could ask questions by placing them in the chat button down the bottom during the presentation, then we hope to have 5 or so minutes of questions at the end of each presentation. Our speakers are both listed here, but I'll introduce them as we go, so we can skip to the next slide then. I just wanted to highlight for those of you who are on the call that you might be quite interested by this particular blog from Holly Ellard, who's looking at communication issues, and the blog is looking at communication issues seen in the genomic medical service by looking at letters seen to their patients. And you can find that on the Genomics England website. And I'll mention this again at the end, but we've got two presentations again in the next meeting, which is of course the last Tuesday of the month. And we've got Claire Shovlin talking about hereditary hemorrhagic telangiectasias and MEK1 inhibition, and Magdalena Bletzer on human endogenous retroviruses. Okay, so now we might go to the first presentation, which is from Dr. Thuy Nguyen, who is a bioinformatician who works within the diverse data team at Genomics England. And she's going to present to us about the effects of ancestry in the 100,000 Genomes Cancer Pipeline. So just again, if as you go, you can put questions in the chat at the bottom, and we'll go to those after Thuy's presentation. So passing over to you, Thuy.
Great. How's my audio? Good?
Excellent.
Cool. And I'm going to share my screen, and let me know when you see it's in presenter mode.
That's good.
Great. And I'm going to start off with saying I'm actually not a doctor. All right. So that's cool. Out of the way. So today we're going to talk about the 100,000 Genomes Project, specifically the cancer program. And we're going to focus on how patient ancestry affected their clinical care while they were enrolled in this program. So the agenda today, we'll look at the program itself, and we'll look at how representative that cohort was of cancer demographics in England. And we'll delve into the clinical bioinformatic pipelines that were used for patient clinical care. So there are two pipelines, and we'll talk about the ancestry effects in both. First off, we'll look at the somatic variant prioritization pipelines. And secondly, we'll look at the germline versions. Right, so ethnic representation. You'll find that in a lot of journals and publications, the words ancestry and ethnicity are used interchangeably. So for this talk, we'll be very pedantic and very strict about what they mean. Whenever we refer to genetic ancestry or just ancestry in general, we're going to refer to the genetic similarity of someone to some reference population. And when we say ethnicity, we're going to refer to a self-identification of some cultural affiliation. And so the main thing that you need to take away from this is that ancestry is something that you need to genetically test for, and ethnicity is something that you have to ask for. So this is the ethnic and ancestry compositions of the 100,000 Genomes Project Cancer Program. On the left is ancestry, in the middle is ethnicity. The first thing that you notice is it's a whole sea of blue in all the plots. So we'll focus on ancestry first. So the majority ancestry is European. But what you also might notice that there's a lot of gray, and those are unassigned. So there are problems with genetic ancestry assignments in this study and a lot of other studies. So as we mentioned before, it's genetic similarity to some population, but that population is arbitrarily defined. And in this analysis, we've used the populations as defined by the 1,000 Genomes Project. And you'll find that a lot of other analysis does similar. So the problem with this is that the 1,000 Genomes Project has 5 major continental populations as listed here. And if a patient doesn't happen to fit within one of those populations, they'll be unassigned. And Similarly, if a patient has mixed ancestry, they also can't be assigned any single ancestry. So you'll notice that patients that are unassigned comprise the second largest ancestry group, which is not an ancestry at all. So in the middle over here, again, you'll see another sea of blue, and this means that the composition of the cancer program is majority white ethnicity, self-reported ethnicity anyway. And we know that UK as a whole is majority white ethnicity. What we wanted to know was whether the cohort had any under-recruiting of minority ethnicities into this cancer program. So we have some of the results here. We compared the 100,000 Genomes Project to the statistics from England, gained from the Public Health England, and this was reported in Delon et al. So I'm going to highlight some things over here. Each subplot represents a cancer and a sex. We're going to look at this point over here. This represents the incident rate ratios in bladder cancer for Black to white ethnicity, as reported in England. So you notice this pink dot over here is under 1. This means that the rate ratio is lower than 1. And this means that Black ethnicity is represented at a lower rate than white ethnicity in bladder cancer in England. So one thing that you'll notice all across the board is that typically minority ethnicities have cancer incidence rates lower than white ethnicity. And this is all across England. In the 100,000 Genomes Project for bladder cancer, the same plot over here, this is a blue dot. And you notice that this rate ratio is higher than 1. So this means the opposite, that Black ethnicity is represented higher than white ethnicity in bladder incidence rates. So this difference is significant, and we've demarcated it here with 3 asterisks. If you look at across all the cancers, you'll notice that you won't see a lot of significant difference between the 100,000 Genomes Project and England itself. And whenever you do see a difference, you'll notice that the minority ethnicity is actually at a higher proportion than you would expect in England. So this happens in bladder cancer, as we just shown, breast cancer here, and prostate cancer over here. So we're looking at Black to white ethnicity ratios, and you'll see something similar when you look at Asian to white ethnicity ratios. So Asian ethnicities are represented higher than white ethnicity in breast cancer. So one caveat that we need to take away from this is that minority ethnicities are represented at low sample sizes in a lot of these cancers. All throughout these plots that we showed, we've only showed cancers with more than 100 patients, but there is a possibility that the sample sizes are too small, that we just don't have the power to detect differences between England stats and the 100,000 Genomes Project. But whenever we do have the power to detect any difference, it is a good sign that the 100,000 Genomes Project does not show under-recruitment of the minority ethnicities. So those are good signs. For the cohort composition itself, we're going to move on to the clinical pipelines themselves. So the basic workflow of the 100,000 Genomes Project was that patients would have their whole genome sequencing, and the genetic variants would be put through this bioinformatic pipeline that would pick out the variants that would be of most clinical use and prioritize them, send them in a report to the clinicians that would be later reviewed and used in patient care. So we'll start off with the somatic variant prioritization pipelines first. This is the workflow, and it's fairly wordy, but I'll let you know what the main takeaways are. Within somatic variants, there are 3 classifications that are important, and they're in descending priority. So the first off is domain 1. These are the highest priority of somatic variants, and these must go to clinicians for review, and clinicians are mandated to review them. And they're lower priority 2 and even lower priority 3. And somatic variants that are of no clinical use are not domained at all, and they're never sent to a clinician. So with domain 1, these are all the variants that have something actionable with regards to clinical care. For example, it might determine diagnosis or prognosis, or it might alter treatment depending on its existence within a patient tumor. So we do notice that there are indeed ancestry differences between the number of domain variants that are sent to clinicians. So we've broken it up by domain 1 over here on this side. In all of these plots, these are histograms of the total domain 1 somatic variants per patient, and we've broken it down by ancestry. So right off the bat, you'll notice that it's really hard to detect any ancestry differences. If you start adjusting for things like sex and cancer, you'll start noticing some differences in that South Asian ancestry has significantly fewer domain 1 somatic variants sent to the clinicians than Europeans in the blue. But if you start adjusting the model for additional covariates, things like tumor mutation burden, that is, and age, you'll start noticing that these ancestry effects start to diminish. And it's the same when you look at all domains altogether. So this is domain 1, 2, and 3. If you disregard any covariates, you will see ancestry differences. But once you start taking into account tumor mutation burden and age, these ancestry effects are no longer evident. So one thing to note is that non-European ancestries are typically younger, and advanced age is associated with higher tumor mutation burden, and that in turn is also associated with more domain variants. So you'll see that the non-European ancestries, they're younger and they'll have fewer domain variants sent to the clinicians. So that's the somatic pipelines. So you'll notice there that ancestry effects existed, but they were more due to tumor characteristics. We'll look at the germline pipelines now, and you'll notice somewhat of the opposite. So this is the prioritization workflow for germline variants. And again, the main takeaways are the classifications. So germline variants are prioritized based on how useful they are in clinical care. And for germline variants, they would be prioritized based on how predictive they are of family susceptibility to cancer. So that would indicate perhaps adding preventive care. And tier 1, these are the most important, these are the highest priority. Any tier 1 variant must undergo clinician review, and any variant that isn't of clinical use isn't sent to the clinician at all. So one thing that you'll need to take away from this is that tier 1 is determined as tier 1 if it's annotated in ClinVar as pathogenic with a lot of research support. Either that or it has to be loss of function through protein truncation. And tier 3 would be classified as such. With this proviso, it has to be a low frequency variant. And the reasoning is that common variants would most likely be benign and not worth sending to a clinician. And this is important to remember later on when we look at the results. So let's start off with tier 1. This is the left panel over here. We're showing the percentage of patients with at least one tier 1 germline variant. These are the highest priority that must be reviewed by clinicians. One thing that we'll note when it comes to germline variants, especially tier 1 germline variants, is that they're really rare. Most patients don't have any. You'll look at African patients here, African ancestry patients. Only about 5% of them will have any tier 1 germline variants. And if you look across all across the board, you'll find that the most any patient will have is one tier 1 germline variant. So that said, we do notice that there's some ancestry differences between having a tier 1 variant or not, in that African ancestry and South Asian ancestry and pretty much all the non-European ancestries are more likely to have at least one tier 1 germline variant. If you start delving into the other factors that might be affecting this, what you'll find is that ancestry isn't as predictive as much as age. So here is the opposite of the somatic world, in that the younger you are, the more likely you are to have a tier 1 germline variant. And this is more of an indication of how we've classified these variants, because a lot of these variants are pathogenic and deemed as such because they confer early age. Now, if you go into the tier 3 germline variants, there is a very, very strong ancestry effect here. Just looking at the histograms, not taking anything into account, you'll start to notice that all the non-European ancestries have more tier 3 germline variants. If you start adding additional factors to account for this, sex, heterozygosity, total mutation, cancer, it doesn't matter. The ancestry effect is still significant and it's very strong. And the largest association that you'll have with having a tier 3 variant is whether you are of non-European ancestry or not. Right, so we looked at reasons why, and one of the main culprits is the allele frequency filtration. So I mentioned before, to classify a variant as tier 3, it has to be considered rare, at least in a cancer context. The problem is the allele frequencies were drawn from a large population, without ancestry stratification. So this is problematic because this population is majority European. And as you would expect, variants that may be rare in European may be actually quite common, say in African ancestry. And this is what we're showing here in this plot. So we've broken up the allele frequencies on based on ancestry, and these are all from Nomad. So each plot shows all the tier 3 variants as a histogram of allele frequencies. So here we're showing all the tier 3 variants in European ancestry allele frequencies. And this black line over here is one of the thresholds used in the filtration. So there are quite a few thresholds used, and we're showing the least stringent threshold. The pink shaded area indicates all the variants that would be thrown out using this threshold. So you notice that in European ancestry, all of these tier 3 variants are considered rare using this threshold. This is African ancestry over here. You'll notice all of these tier 3 variants that made it past the threshold in the original pipeline and made it into a clinical report that was sent to a clinician. And you'll notice this variant over here, it happens to be around the 10% frequency mark, which sounds pretty common and is probably benign. So this is African ancestry. And if you look at all the variants that shouldn't have made it past this threshold in African ancestry patients, it accounts for roughly about 20% of the variants that shouldn't have made it to a clinician at all, but did. And this is within the tier 3 classification of the cancer pipeline. And there are other parts of the pipeline that use allele frequency filtration. For example, flagging germline contamination. So this is one fix that we can do for the pipelines in future iterations. So one thing that I forgot to mention is the 100,000 Genomes Project served as a foundational model for the GMS. And these pipelines are still in use in the GMS right now. And this is a good option to fix the pipeline for better equitable healthcare between all ancestries. Right, to sum it up, 3 main takeaways. First is that the 100,000 Genomes Project Cancer Program is probably representative of England, and there wasn't any sign of under-recruitment of minority ethnicities. The second is that you'll find differences between ancestries in the somatic variant prioritization pipelines, but that'll be down to tumor characteristics. And the third one is the most important one. You also see ancestry differences in the germline variant pipelines, but these differences are due to bias within the pipelines themselves. So one thing that I'll note is that we mostly focused on the clinical bioinformatic pipelines here, but it's really worth looking at the downstream effects on patient clinical care, in particular in the germline arena. For example, clinicians may order additional orthogonal validation for any variants that they deemed of clinical use from these reports. Things like PCR or Sanger sequencing. So it is possible that non-Europeans incur additional healthcare costs because of these false positives that are being sent to clinicians. And in general, we encourage people in our organization and all other organizations to include ancestry and ethnicity into their analysis because this is the first step for equitable healthcare. And thank you. That's it for today.
Okay, so thank you very much, Thuy. There are questions coming up in the chat, but I'm going to take chairman's prerogative and ask a couple of questions in the first place. So one thing I'm always interested in is whilst we divide up Asians into South and East Asians, and I know in our rare disease analysis there are big differences between West and East Africans, and in fact Africa is hugely diverse in itself. Why do we not break up Africans into other sort of finer scale ancestries, or do we actually have plans to do this? Have you looked at that, the effect of that?
Yes, if you'll stay tuned to other research seminars coming up in the future, the data diversity team from Genomics England will show the analysis in the rare disease cohort in which the ancestries are much more granular, and you'll be able to see the differences between East Africa and the rest of Africa, and it is stark. So we have not done it here. We've decided to use the original 1,000 Genomes Ancestry Assignments. So these are currently available with the NGRL, and they're readily available for everyone. And they were the first ancestries that we started with. we looked at more granular ancestries, and we found that, unfortunately, with the cancer cohort, which is significantly smaller than the rare disease cohort, it's a lot harder to tease out the differences between the different ancestries because of the sample sizes. So it was an issue of power. So for this analysis, we've decided to maintain the original and slightly subpar population definitions.
Okay, and can I just ask, I can understand in rare disease analysis why having an excess of tier 3s would be problematic, because it makes it much harder for people to actually come up with a diagnosis. But for cancer, given that those tier 3s, my understanding is that they don't have a known clinical implication, what difference is it actually likely to have made to a patient's care?
So typically, clinicians would most likely just look at the tier 1s, and there are very few of them. So it is not an issue of, I guess, having too large of a clinical report to validate. So improving the tier 3 will probably have a lesser impact than improving the quality of tier 1s. That said, because of the lack of research in non-European ancestries, some of those tier 3s might actually be tier 1s. And if you had a better list of tier 3s, you might have a better chance of detecting the pertinent variants for clinical care, for patient care.
Okay, all right, thank you. That's very helpful. Then I'm just going through the questions that are in the chat. So question 1 is about variant prioritization. So it's about, why is variant prioritization by ancestry, especially between germline and somatic? I think you've explained why germline variant prioritization is by ancestry, but what about somatic?
At the moment, the original pipeline for the 100,000 genomes and the GMS pipeline in place don't consider ancestry in the cancer pipelines. However, for future pipelines, things like tumor-only pipelines, they are looking at ancestry allele frequencies because this hugely plays into whether you throw out a variant or not.
Okay. All right, so there are a couple of questions about how you actually called ancestry. One is a technical question about PCA or admixture and how many SNPs, and the other one based on, I think the second question sort of confuses ethnicity and ancestry, but perhaps if you could just basically describe for us how you've actually assigned ancestry in this group.
Yes, it's a comic technique, and I think it was used in the 1,000 Genomes Project and various other studies as well. So it's something that almost everyone uses when they talk about ancestry. But it's a random forest classification based on genetic distance from principal components. Cool. And the principal components are based on, the 1,000 genomes.
Yeah. Okay. All right. There's a question here about the explanation for why you have fewer prioritized somatic variants that you ascribe to having younger patients, which I'll come back to too. But a question is whether that's because there's actually just less known about cancers in non-Europeans and therefore fewer tiered variants.
I think in the somatic pipeline design, the opportunities for ancestry bias are less so. So in the somatic pipeline, we're looking at the entire gene as a whole. So there are certain genes oncogenes that are related to cancer, and any variant within that gene would be thrown into the domain classifications, either 1, 2, or 3. So we're not looking at any particular variant that would be more biased towards one ancestry than others. And yeah, so I think in general, younger patients are less likely to have a higher tumor burden, mutation burden. And because of that, the sheer number of tumor mutations going into this pipeline for prioritization will be fewer. And so there will be fewer coming out. Okay.
So can you explain to me why you think why we ended up with younger patients in the non-European ancestries? Is that just an effect of the age of non-European ancestry groups, or in fact ethnicity groups even in England?
My apologies. I used to have a slide for this, but I don't have it today. But in England, UK as a whole, and various other Western countries, you'll find that the minority ethnicities non-white ethnicities will be at a younger age demographic than the white ethnicities. And so consequently, European ancestry will be older and non-European ancestry will be younger. And this is just typical of the country demographics.
Okay, so then there's a question about NOMAD as a resource for getting allele frequencies for somatic cancer variants. The question is that the best one? So I'm thinking you used it for germline, but did you use NOMAD in some way for somatics as well?
So when we were showing the NOMAD slide, we were showing hypotheticals. So this isn't actually applied to the pipeline. We're showing what would happen if you were to apply it to the pipeline. But yes, the allele frequencies that are used in the actual pipeline are based on an early subset of the 100,000 Genomes project. So it's not the ideal subset to obtain allele frequencies from. And if we were to apply this, we would be using it on germline. So they would, whether they have any relation to cancer, it would be irrelevant. We're simply looking at whether a variant is common or rare within the population.
Okay. So I was also thinking about the differences in, the covariates that you actually applied to it. And you had several smoking-induced cancer types in there, but I didn't see an environmental risk factor like smoking, which does vary by ancestry in the UK. Included? Do you have data for that?
We didn't use smoking in particular. We used things like socioeconomic deprivation, and we found that that had very little impact in our models, and so we took it out.
Okay.
But we use proxies for things like smoking, like tumor mutation burden. So things like lung cancer, where there's a high tumor mutation burden, you'll expect more domain variants coming out of that simply because there's just mutation happening in any oncogene itself.
Right, cool. So can we come back to the question of the comparison of your pipeline with the current GMS pipelines? Is there a difference in the actual analytic approaches, or is it mainly about the somatic variant databases and germline databases that people are using to interpret the findings?
The databases are more or less the same. I mean, there have been upgrades to the databases since the 100,000 Genomes project. That said, so it's something that we haven't presented here. The databases of pertinent genes, I think we found no issue with at the moment. But we did find issues with variant databases, specifically ClinVar. So other studies have noted that I think even the 1,000 Genomes project noticed that ClinVar has, a certain amount of ascertainment bias in that most genetic research is done in European ancestry. So most pathogenic variants will be in European ancestry. So you'll notice a very slight, but not very significant European bias even within our data set.
Okay. And these biases, I'm presuming that most of your analysis is focused on single nucleotide variants. Does it apply equally to structural variants and copy number variation?
Most likely, yes, but we have not done full analysis on any types of structural variants, including CNVs. So I'm reluctant to give a positive answer
about that. Okay. All right. Look, that was really excellent. Thank you very much, Thuy. I'm sure you'll be a doctor in the near future if you keep going this way. All right. Thank you. All right. So for the second presentation today, we have Tembi Fashina, who's a PhD student, and she's going to present to us. There was an error in the original slide that we put up, but it was correct that she is talking about investigating microRNA-associated variation in the epileptic encephalopathies. Tembi, over to you. Thanks, Thuy.
Thank you very much. Can you see this okay?
Yes, and it's in presentation mode too.
Perfect. Good afternoon, everyone. My name is Ifeolutembi Fashina, also known as Tembi. I'm a fourth-year PhD student at the Royal College of Surgeons in Ireland, and I'm also under the Science Foundation Ireland Centre for Research Training in Genomic Data Science. So my talk today will be part of my PhD work, which is investigating the role of rare microRNA-associated germline variants in the epileptic encephalopathies. So it's a mouthful, but I'll try and make it reasonably understandable. So I'll first of all talk you through the genetics of the epilepsies for those who are not familiar, discuss microRNAs and how we look at variation within microRNAs, the objectives of my work in Genomics England's infrastructure, my methods, and then my finding in 2 different cohorts. So to start off, epilepsy is a brain disease characterized by an enduring predisposition to generate epileptic seizures. We used to think of epilepsy as one disease, but we're starting to understand or see it as a constellation of diseases. So if you look on the right-hand side of the slide, you see a graph where we have acquired epilepsies on the extreme right, and then we have genetic epilepsies, where we have polygenic inheritance in the middle, and then single gene epilepsies on the left-hand side. And my work focuses on the single gene epilepsies, which are the rare severe epilepsies in comparison to the common epilepsies, which would often be polygenic. And in particular, I'm looking at the epileptic encephalopathies, which are rare severe genetic epilepsies of infancy and childhood. And again, in context of that constellation, within the epileptic encephalopathies, which I'll be calling the EEs, there are multiple subtypes and, let's say, sub-diseases within that umbrella. So the genetic architecture of the EEs is primarily monogenic of dominant inheritance. Excuse me. Okay. In GEL's Panel App version 3, that's a bit outdated now, there are about 540 confirmed epilepsy genes. So these are within the green list. And that number has come about due to an expansion in the number of epilepsy genes over the next generation sequencing era. So if you look at the chart on your left-hand side, you can see that exponential growth in the number of genes associated with the epilepsies. And that 540 includes monoallelic and biallelic genes. Now, often the pathogenic variants within these genes are de novo and exonic. So they're de novo, they're not usually seen in either parent, and they're exonic, they're usually found within the coding regions of the genome. Now, we've established that de novo pathogenic coding variants can often cause EEs, but we're starting to see evidence due to work of different large consortiums that inherited rare coding variants can also confer some risk in the EEs. And when we look at this chart on your right, you'll see that we are sitting somewhere at the top left in the brown box, where we're starting to see that rare alleles can drive Mendelian disease in the epilepsies, which is something we haven't really seen before. But for common epilepsies, we would be around the, sorry, let me find my laser. Oh, we'll just ignore that. For common epilepsies, would usually be around the common variants implicated by common disease by GWAS group there. So the way we study these rare variants is through gene or region-based rare variant analysis, which is a different method than we use for de novo analysis or GWAS. And the findings from these rare variant association analysis in the EEs and rare epilepsies is that ultra-rare damaging coding variants, especially protein truncating and damaging missense variants, are enriched in cases compared to controls. But we haven't really studied damaging non-coding variants. And part of the issue there is defining what damaging non-coding variants are. So altogether, for now, you just need to keep in mind that de novo and inherited ultra-rare damaging variants have been found to play a role in the EEs, and there's a possible role for ultra-rare non-coding variants. But in particular, I'm interested in a specific type of non-coding regulator called microRNAs. And these are 22 nucleotide long RNA sequences that are transcribed from DNA. The schematic diagram on your right-hand side shows the seed region highlighted in the brown box of a microRNA. And that's the bit that is expected to bind to the mRNA at its 3' UTR. And microRNAs translationally, well, they repress mRNA expression or allow their degradation. And there are over 2,000 microRNA genes in the human genome. More importantly, microRNAs can be conserved across multiple lineages or non-conserved. And non-conserved microRNAs have been implicated in human brain evolution, which is quite important and interesting when we're talking about epilepsy, which is a brain condition. So altogether, there's evidence of microRNA dysregulation in the epilepsies, and we're now trying to see how that plays into the rare epilepsies. Outside of the microRNA, we need to discuss the targets of the microRNA, which is the 3' UTR of the messenger RNAs. The schematic on your left highlights the 3' UTR. And while the 3' UTR is the primary or canonical target of microRNAs, it also regulates the mRNA through other processes, including polyadenylation, and it also has sites for RNA binding proteins. So we suspect that variation within the 3' UTR could affect A, microRNA binding ability, and B, affect other regulatory functions. So altogether, in my work, I refer to microRNA-associated variants as those within the microRNA itself or within the 3' UTR of their targets, excuse me. So the objectives of this part of my work the hypothesis here is that rare damaging microRNA-associated variants are enriched in the EE cohorts compared to the controls. And I'm going to assess this using region-based rare variant enrichment analysis through the whole genome sequencing data that I've accessed in GEL. But for this talk, I'll just be focusing on the enrichment of conserved 3' UTR variants within confirmed epilepsy genes, and we'll discuss this in more detail as we go on. So the first thing I did was to identify my cohort within the 100,000 Genomes Project using the research environment. So this is within the rare disease cohorts in contrast to, say, the cancer cohorts. And we have, again, access to whole genome sequencing data from all the individuals that have been recruited in this cohort. I identified 318 people who were recruited on the basis of having a diagnosis of the epileptic encephalopathies, and then used 1,233 individuals with cystic kidney disease for the purposes of using a non-neurological control, using SQL and RLAB key in the research environment. After identifying our individuals of interest, I went into GEL and looked through the QC that had been run. So that first line that says GEL analysis is just the quality control that had been done on the variants that were submitted or that we can access in GEL. And we have access to over 540 million variants that pass quality control steps. For the study level quality control, I took just those variants that passed GEL's own QC, looked for variants with high quality calls, and looked specifically at single nucleotide polymorphisms, that's SNPs. After getting those variants that passed QC, then I merged my variants of interest for cases and controls so I could study them. You'll see why in a few slides. And then re-annotated using Revel, Polyphen, Lofty, and Nomad. The reasons for those annotations will become clearer again. But most importantly, I filtered just for variants within the green list from Panel App, and I looked for variants that are absent in Nomad, with the rationale being that variants that are absent are not well represented in our populations because we're looking for ultra-rare variants. So following all that preparation, then I did genotype extraction for my variants of interest for each individual and ran ultra-rare variant burden and collapsing enrichment analysis. So in regards to the results, the first thing I'm showing you here is the gene set burden in the coding region. So this is our sanity check, because if you cast your mind back a few slides ago, I mentioned that we have started to see evidence that ultra-rare variants in the coding region are enriched in cases compared to controls. So we're just trying to ensure that we could see the same effect in the GEL cohort. So I ran a logistic regression here using case control status based on the presence of ultra-rare variants in any of the 540 epilepsy genes, and using sex and the first 5 principal components as covariates. So that's supposed to account for population structure. So this forest plot here shows you the burden across cases and controls for each functional class. So I'll just like to point out the 2 points in purple here, where we can see a significant enrichment of loss of function stringents. That's just the way these loss of function variants have been annotated. Significant enrichment of damaging missense variants in cases compared to controls. And most importantly, when we look at the synonymous variants, we don't see any enrichment of those loss of function or damaging missense variants. So our sanity check is working. And what we then wanted to move to was what we don't know, which is, say, looking at functional classes within the 3' UTR. So today's talk mostly focuses on conserved 3' UTR bases. And the first thing I needed to establish was the conservation threshold for ultra-rare variants in the 3' UTR relative to the CDS, where we know that the coding region is often under tighter regulation, often under tighter conservation than the 3' UTR. So these histograms here are not on the same scale, but we can see that there are ultra-rare variants that are strongly conserved in the coding sequence. And we know that because the higher the Philo-P score, so this is the Philo-P 100-way score, the higher the level of conservation. And we can see that there are also some highly conserved 3' UTR bases. And so we split those 3' UTR bases into 4 conservation bins to identify extremes. And we ran our logistic regression again. And so what you're looking at here is another forest plot where Q4 represents the most conserved bases within the 3' UTRs of our 540 epilepsy genes. Q3 is less conserved than Q4, Q2, and so on. But Q1 is missing here because it's not informative. And so looking at this forest plot, you can see that there's an enrichment for Q4 3' UTR variants in our cases compared to controls. We then wanted to see what genes were driving this enrichment that we see. So we wanted to look at the coding variants and the 3' UTR variants side by side. And this was just done through Fisher's exact test and QQ-perm, which helped us estimate the expected p-values. So on this slide, on the left-hand side, we are looking at the loss of function and damaging missense variants that are contributing to the signal in the coding URVs. And on the right-hand side, it's the Q4 3' UTR variants. In purple on the left are our general epilepsy genes, while the ones in green are specific to the EEs. And so we can see that, of course, we only use the epilepsy genes here, so that's not too shocking that it's just epilepsy genes. But we can see that the lead gene here is a general epilepsy gene, but we can see one dominant EE gene in the damaging missense and loss of function ultra-rare variant burden testing. On the right-hand side, we can see that SCN2A, which is specific to the EEs, is one of our lead variants. So we did some power calculations to see how we were performing, and the green line here shows the corrected p-value threshold that we'd be expected to meet, and we are obviously way below that. So our study is underpowered, but the framework holds. We then moved on to our larger cohort, and we did this by including individuals with neurodevelopmental disorders. And so again, using the same tools, RLabKey and SQL, we knew or know that there's a shared genetic element between the epilepsies and a number of neurodevelopmental disorders. And so what we did was to identify individuals with a seizure, HPO, and any of the other 7 that are listed below. And so we got this larger cohort of 1,560 cases and 1,233 controls, the same number of controls. When we run our logistic regression again, we see that our synonymous variants are still insignificant and still are around the odds of 1, but we see a much better odds for our loss of function and damaging missense variants. And we still see that that enrichment for Q4, so the most significant UTRs, most conserved 3' UTR bases within the 3' UTR. And so the genes that were most responsible for these signals, there's supposed to be a QQ plot here, but I need to figure out some technical issues. The genes that are representing the signal are SCN1A, SCNH8, KCNT1, and RGDI. The next question then would be, what are the possible effects of these conserved 3' UTR variants? And we're looking at that in 3 ways. The first is to see if the variants are affecting any polyadenylation signals. So if you just remember this schematic here with the polyadenylation poly-A tail at the end of the 3' UTR. We used a tool called UTR annotation, and that essentially tells you if the sites of the polyadenylation signals within your mRNA and your transcript, and to see if there are any changes in those sites due to the presence of your variants. And so far in our genes of interest, I haven't identified any changes in polyadenylation signal. So I'm now looking at estimating changes in mRNA stability through RNA fold. And then I'm going to use target scan to predict changes to the microRNA binding ability of these mRNAs. Excuse me, that's a typo. So altogether, I've implemented a framework for ultra-rare variant burden and collapsing analysis within the 3' ETR and the coding region. We've replicated the expected results in the coding region in 2 cohorts and identified a novel signal for 3' ETR bases in the 2 cohorts. And what I'm doing right now is trying to establish how the conserved 3' ETR variants affect mRNA function. I'd like to thank you all for listening. Thank Genomics England's consortium and patients, as well as the team that helped with technical issues, my supervisors, and my funders. I'm happy to take any questions.
Okay, that was a really clear presentation. Thank you, Tembi. So again, people, if you could put questions in the Q&A box down the bottom. I'm all going to start off with Chairman's prerogative again and just ask about so validation of these, and in particular, sort of downstream functional validation. I wonder if you're aware that Genomics England has a large RNA-seq data set that might be useful for this, and we're generating also a proteome data set, and wondering if you've thought about using those for validating your findings, and particularly trying to get from group level studies, as you're doing at the moment, as you're sort of burden testing to individual findings.
Yeah, absolutely. That's a great question. And yes, I've reached out to the team that gives permission for the RNA-seq, and I currently don't have access, but it is something that we've considered because it would be the best way to see if we can see those transcript level differences in our individuals. Yeah.
Yeah, I'm also thinking that given that you're particularly interested in microRNAs, and of course they won't turn up too much in the total RNA set, but if there are specific individuals that you might wish to be followed up, then that's something we could consider as well.
Yes, for sure.
Okay, so moving over to the question. So question 1 also looks at follow-up studies. So that's replication studies. Are there large data sets out there that you can do replication with? And have you got any plans to do so?
So far, we have access to, or could have access to, some data from EPI25. But the issue there is, again, we might be restricted to the exome. So we want whole genome sequencing so we can actually look in depth to those 3' ETR variants. So that's definitely a big limitation at the moment.
Okay, all right. So there's a question from Patrick Campbell, which is saying to essentially think big and go beyond epilepsy. What about just generalizing this research across rare disease probands and linked disease genes? So basically all diseases, for example, that are in the green panel app.
That would be great. I know that there's a team working on 5' ETR variation in gel. That is beyond the scope of my PhD, but again, it would be pretty exciting, you know, especially when we get the validation work working. It would be, there'll be more rationale to then follow that up in, you know, a larger disease group. So
right. So you're in year 4 of your PhD?
I am, yeah.
If I was your supervisor, I'd be telling you not to do this, but it sounds like a really good postdoc project. All right. Then there's another question about did you combine dominant, recessive, and X-linked genes together for the burden analysis? So you commented in the first place that these were predominantly, I guess, de novo dominants. Yes. Do you see many other recessives or X-linked genes, and have you analyzed them all together or not?
Okay, so I did start off with everything together. I didn't show you the results for just the dominant genes, but you do still see the same effect if you look at just the dominant genes. I've been doing some more analysis on a different question, but for this bit of the work, yes, you still see the effect if you keep it just to the dominant epilepsy genes.
How, just interestingly, statistically, how did you cope with dominant versus recessive versus X-linked genes?
In what sense?
Well, in the sense, did you just count it effectively as that somebody is it treated them as all as dominant, or did you do it by allele counts? And how?
So fundamentally, I took the presence of one, how do I phrase this? Yeah, so I just took anyone that carried the variant, whether it was 2 counts or 1 count, I took that as presence. So that's just the way. It's more of a, yeah, it's a collapsing analysis there.
Okay. All
Yeah.
right. Okay. So then you actually commented about statistical power, and there's another question from TM also asking about, do you have enough samples for power analysis for rare variants? You commented that you didn't. So how many do you think you actually need?
So for the EE cohorts, that's the first one I showed you. Keeping that ratio of cases and controls, we needed about 500 cases to even bring it up to a level where we could adjust for just the tests on the coding region. So sorry. And then but when, yes, sorry, someone's at the door. But when we then looked at the larger epilepsy cohort, we seem to be powered enough, but I need to go back in and do that again. So we might be able to reach the degree of power we require for the coding regions, but because we're testing the coding regions and then the 3' UTRs, we're essentially going for a more stringent analysis then. We're going to have to correct for all of those together, and we probably won't reach that.
Okay, all right. So just coming back to Patrick Campbell's suggestion, I'm presuming you've heard of Thuy comment, which is generally used in genome-wide association studies. You find an association and effectively you fish to see whether any other phenotypes, say, or diseases actually are associated with it. Have you tried or considered trying a similar thing with burden testing?
I actually have not, no, but I could look into that.
Yeah. Okay, because then I think it is sort of aligned to what you were doing with the schizophrenia enrichment analysis, that you could sort of look across diseases that way and see if you pick up any more evidence to see that these are functional.
Yeah, I'm not sure how it works for rare variants, but I'll definitely have a look to see.
Okay, so then there are a couple more questions that are coming up in the chat. So Matt Silver asks, do you have any candidate microRNAs that would allow you to do targeted analysis with relevant microRNA binding sites? So I guess, can you take this to an individual level and do functional analyses on these?
In principle, yes. So the way I'm currently looking at the microRNAs is by their evolutionary specificity. So I'm going human-specific microRNAs, primate-specific microRNAs, and then others. And the idea is that if I'm able to find variants in the seed region of one of these microRNAs, even if it's in like 5 individuals, but it's pulling in, then we should be able to follow up and do some actual work in the lab. I say we, not me in particular, but we, yeah.
Okay, all right. So, and then there's an excellent question. Did you use the synonymous variant analysis, negative control, for your burden analysis in the study?
Yes.
Okay, cool. That was a simple and quick answer. Then somebody else, I think, is largely congratulating you. How do you combine mRNAs and data science? Followed by a round of applause. Okay, I think at this point, I think we've exhausted questions and we should stop there. Tembi, thank you so much for that really excellent presentation and answering those questions. Which case we've come to the end of the seminar. Just to remind everybody that we've got the next seminar is on the last Tuesday again, so February the 27th at 2 o'clock. And I look forward to seeing everybody on that at that slot. Thank you then.